and I'm interested in the evolution of Salmonella enterica as a pathogen of livestock. So Salmonella enterica is an incredibly important pathogen, causing an estimated 1.3 billion clinical cases of gastroenteritis per year. The pathogen mediates all levels of the food chain and can even be acquired from companion animals. Salmonella enterica can be divided into 7 subspecies. Subspecies 1 can be further divided into almost 1,500 different serovars. It's at this serovar level that we notice differences amongst host associations. So some serovars have a very broad host range, while others have a very narrow host range. It is the aim of my PhD to identify and characterize potential mechanisms that can maintain these distributions. So to do this, I decided to compare 2 serovars that had completely distinct host associations. I identified these by looking at isolation statistics from the UK and the USA, with the assumption that if the isolation pattern was the same between these 2 geographically isolated locations, that it was a property of host-pathogen interaction. So using this criteria, I identified S. darby, which is predominantly isolated from pigs and turkeys, and S. rebendeka, which is predominantly isolated from cattle and chickens. So to start my research, I decided to sequence the chromosome of a few strains of each serovar. I performed functional annotation using the raster annotation system, and by comparing these functional genomes, I managed to identify potential phenotypic differences, for which I could then develop phenotypic tests to study. I also performed a basic sequence alignment and discovered a new Salmonella pathogenicity island, SPY23, in the chromosome of S. darby. So the results of comparative functional genomics showed 3 major domains of phenotypic variation between the 2 serovars. The first was in metabolism, and I further studied this using biolog PM technology. The second was in persistence and resistance, and I studied this using assays such as bioformation and antimicrobial resistance assays. And the third was pathogenicity, and I studied this by further characterizing SPY23. This is what I'm going to focus the rest of the talk on. So SPY23 is 37 kb in length and contains 42 open reading frames. Now, the island is found in various serovars of Salmonella, but the gene complement in S. darby is completely unique. You can see in this diagram here that the links represent nucleotide sequence homology, and you can see that darby, from about the center to the very end, is completely unique. This represents 17 unique genes. So as I said at the start of the talk, Salmonella darby is predominantly isolated from pigs and turkeys. Now, a high proportion of these isolations are actually from pigs, so I decided to study the expression of this island in relation to porcine colon and jejunum explants. I did this using the iBox system. I then isolated the RNA and performed qRT-PCR. You can see in this graph here, this is the expression level relative to a no tissue control. And you can see that the island is upregulated in both tissues. Yet the island is upregulated to a greater extent in the jejunum, as those bars marked in blue. The 2 most upregulated genes were DOCB, the gene on the right. This is a putative type 3 effector protein, and it's found in every SPY3 that I've looked at to date. And POTR, which is a gene that's unique to the SPY23 from S. darby. And this gene has a lot of sequence homology with PILB, which is found in S. agona. This gene allows the production of detachable pili on the cell surface. So I decided to study this through allelic exchange mutagenesis. So upon initial culturing of the mutant, it was indistinguishable from a wild-type strain until the cultures were left to sit on a surface for a period of several hours. You can see in the mutant strain that the cells were agglutinated into one cohesive cell mass. And it's also at this point that I noticed the mutant was susceptible to salt and cold temperatures. So if we take an equal volume of these 2 cultures and take them out onto LB agar, and then incubate them for 24 hours at 37 degrees, we get 2 distinct colony morphologies. You can see that the mutant strain forms these much larger yet fewer colonies. And the mutant is also surrounded by this translucent halo, let's say, whereas the wild type is opaque to the very edges of the colony. So this agglutinating phenotype made enumeration through serial dilution impossible, so I decided the best way to further characterize this strain was through using microscopy. So you can see here 2 negative stain images, and you can see clearly that the POTR mutant is covered in pili, whereas the wild-type strain has very few pili and few type 1 fibrilli. So how did this contribute to pathogenicity? Well, as I just showed, the gene POTR was most heavily upregulated in the jejunum. So I decided to look at how pathogenicity was affected when exposed to a porcine jejunum-derived cell line. Sorry. So here you can see differential stain confocal micrographs. The blue circles represent the porcine nuclei. The yellow represents a bacterial cell that has adhered to the surface but not invaded. And the red represents a cell that has invaded the porcine monolayer. And you can see that the muted strain has associated in a lot fewer numbers than the wild type, and that there's no real evidence of invasion. So as I said at the start of the talk, the association between the host range and the cerebrale is obviously at the cerebrale level. So it's integral to understand the population structure of the isolates that make up these statistics. So to do this, I sequenced 7 housekeeping loci. I then concatenated these sequences and produced this phylogeny. Now you can see the isolates in Ethmobabeca formed one large clonal expansion, and all these isolates lack SPY23, whereas S. darby formed 2 distinct lineages. Lineage 1, which is isolated from pigs, contains SPY23, and lineage 2, which is isolated from turkeys, which lacks SPY23. So you'll probably like to ask the question, how does this affect pathogenicity of these isolates when exposed to the porcine jejunum monolayer? Well, isolates from lineage 1 associate with much higher numbers of the monolayer than isolates from lineage 2. And you can see, sorry, those bars marked in orange from lineage 1. So we can conclude that SPY23 is either an indicator of porcine pathogenic strains of S. darby, or that it's a determinant of it. So to address this, I've sequenced S. darby E4 to see if the chromosome surrounding the island is isogenic. Okay, so that's quite quick. So in summary, functional comparative genomics was a very useful tool for identifying potential phenotypic differences, for which I could then develop phenotypic tests. I also showed that SPY23 was upregulated in the jejunum and downregulated in the colon, and that this gene POTR that's found on the island allows the cells to become more planktonic. So in the jejunum, this potentially allows them to cover a larger surface area and infect more cells. And then when they migrate through to the colon, they agglutinate, allowing them to prepare for density-dependent phenotypes such as bioform formation ex vivo. So I'd like to thank my supervisors, Martin Woodward and Vincent Janssen, my PhD advisors, Manal Habowen and Liliana Petrovska. I'd like to thank Bill Cooley and David Everest for performing the microscopy. I'd like to thank ECCMID for selecting my talk, and ECCMID for awarding me the travel grant. And most of all, I'd like to thank you for listening.